Hello everyone. My name is Anshuman, and I work as a machine learning engineer at BenchSci. Before we just get into all the technical stuff, I just wanted to frame the problem that we are trying to solve at BenchSci a lot more clearly. So, in order to do that, let's look at the drug development process, which is both time-consuming and costly, as you saw in the previous presentation. So before the life-saving drugs can even make it to the market or are tested on humans in clinical trials, they undergo a process of drug discovery through preclinical experiments to find the best candidates. And it is normally a three-stage process consisting of target discovery, where the scientists study the disease to understand the biochemical mechanisms and identify a target that can affect those mechanisms. Then they have a process to screen those compounds. This can happen by physically testing those compounds as well as through computer models. In fact, machine learning is starting to play a huge role in generating lots and lots of these novel candidates, as you probably saw in the previous presentation. And finally, we have the experiment design stage where we actually have to validate those candidates. So the thousands and thousands of new candidates that are being generated lead to more and more experiments. However, the problem is that half of those experiments are not reproducible, leading to a lot of waste due to poor experiment design. As you can see, a lot is being done to push science forward in terms of potential drug candidates. Little has been done to make the experiments themselves more efficient and reproducible. And that is essentially where BenchSci steps in. Our mission is to speed up preclinical research phase by eliminating unnecessary experimental expenditure. In other words, we seek to digest all the experimental data in scientific publications and present it to the scientists in a fast, highly specific way, which helps to inform their decisions when they're making decisions to buy reagents from vendors, which are tools that the scientists need to actually conduct a successful experiment. So for example, if I wanted to conduct an experiment using a protein called TGF beta in a mouse brain to study epilepsy, instead of the weeks and months it would normally take me to do manual research and validate that protein, I can just go to BenchSci and find the protein in the specific context that was used in a previous experiment very quickly. So you might be thinking, where does visual perception, computer vision, and machine learning come into all of this? Well, take a look at the figure on the right side of your screen, which is pulled straight from a publication. What do you see? Some graphs, some strange gray blots? Well, I have a scientist friend called Yelena, and when I ask her what she sees when looking at a figure like this, it is not the isolated visual features such as the colors or the blots, but she understands the underlying experiment behind the figure. She can understand the lab protocols, the panels, the experiments, the context, the relationship between the experiments, the meaning of the different signals being quantified here, as well as the underlying bioinformatics, all embedded inside this one figure. And the task for machine learning here is to capture Yelena's domain knowledge by building a layered pipeline that captures the different modalities of perceptual learning that she has built up over years of working in a lab. An analogy to this might come from self-driving cars, which most people should be familiar with. To capture the driver's domain knowledge, the ML models must capture the layout of the roads, trees, buildings, cars, and people. It must also understand the meaning of the traffic signs after reading them, along with many subtle details such as like which side of the road to drive on. Similarly, a scientist has to navigate the layout of experimental results. They must understand the techniques like blots, stains, schematics, as well as panels like what does A, B, C, and D here mean. They must read the text in these images and understand the spatial and biological context of that text. Why is this text here instead of some other place? What does the size of these black bands here represent? And so on. And so working closely with domain experts like Yelena becomes an important step of the modeling process to understand the hierarchies of features that would provide the most value to our users, who at the end of the day are also scientists. And the way we solve this problem is by asking Yelena what she sees in a figure like this and converting those into solvable machine learning tasks. So, for example, she sees various panels and experiment regions in the figure and what they represent. She locates and reads the text, which indicates something about those experiments. She looks at individual experiments which catch her eye, which might indicate a signal which have some underlying meaning related to the experiment, as well as she might want to know the presence or absence of reagents such as antibodies, proteins, etc. And once she has all of this information, she needs to apply some logic or her intuition to stitch together a representation of the underlying biology. And we generally do that by using bioinformatics. Using known relationships and graphs such as protein-protein interaction graphs, which tell us exactly how two proteins interact, we can relate entities found in images and put them in the correct biological context. It allows us to understand the proteomics at a figure, paper, and global level and unearth very interesting relationships. So let's look at a couple of projects that highlights the difficulty of capturing this domain knowledge. The first of which is detection of antibodies, which are a very popular reagent group in these kind of tissue section images. So looking at this figure from a paper, we see that it has quite a few tissue section images in panels A and D. And the task here is to extract each individual region and classify it as containing an antibody or not. Tissue sections or histology images with antibody are called immunohistochemistry or IHC. These are very useful to scientists because conducting a successful IHC experiment is hard and depends on a very, very specific protocol. And by detecting these experiments and the surrounding experimental context, we can provide scientists with a definitive way to reproduce these signals in their own labs. And as you might have noticed, these histology images come in various colors and features. So let's take a look at a harder example here. This image has 4 tissue sections, and only a couple of them have an antibody signal. I don't know if you guys can tell. An image like this is actually really hard to classify even with a human eye. However, my friend Yelena was pretty impressed with the result of our models in this image. As you can see, B and D are the ones with the antibodies. In fact, our model actually performed pretty well for this project with a precision of 94% for IHC images. And so just moving on to some key takeaways from this project. Bottom-up labeling, by which I mean we gave our dataset labeling team, which consists of scientists, a decision tree of 29 subgroups of histology classes distinguished by various visual and experimental contexts, instead of just 2 classes such as positive and negative. This helped us build a model that could map inputs to the respective classes a lot more cleanly and intuitively. In addition, we developed some custom loss functions that helped us balance the classes as well as tailored more towards local features, because as you can see in these kind of images, the antibody signal can be a needle in a haystack to detect. And finally, reiterating the overall idea of this talk, we kept scientists in the loop at every stage of this project, which helped us make some very critical data science decisions. And quickly, the second project that I wanted to share is called Western blot digitization. Western blots are a very popular technique among scientists. They are used to study proteins and how they interact with other molecules by looking at the intensity of the bands or the signal here, which indicate the quantity of the proteins. Converting Western blots to data frames by quantifying signals is very valuable. Not only does it help standardize the quantification process, which up till now involved very primitive softwares or just eyeing the signal, it helps us understand the interactions between protein expressions within a Western blot through the use of bioinformatics graphs called protein-protein interaction graphs. So the goal of this project was to detect the bands in a Western blots accurately and quantify them in such a way that the value is one that matches the expectations of the scientists. And that involved identifying the individual bands, isolating them, applying background subtraction algorithms, as well as some useful signal estimation algorithms that mirror the variables that the scientists might find useful. So the overall output was a structure, a data frame structure such as this. But as you can see here, reading the axis of Western blots is actually pretty challenging. The plus and minus symbols here have specific meaning in terms of the experiments and need to be parsed as such. However, using the domain expertise of scientists again, we were able to build a quantification algorithm in such a way to produce numbers, which you see here, that match the intuition of scientists. And we validated that again and again. And as we begin to quantify these experiments and assigning them numerical values and structure, it allows us to do some pretty cool things with them. Having these images quantified allows us to track the interactions of the proteins within these experiments and confirm those interactions through bioinformatics graphs, which scientists will tell you is of great use. So at the end, to summarize, speeding up reagent selection is a potent application of machine learning technologies. It helps us make experiments efficient and reproducible. As part of that pipeline, computer vision models need to be aligned with domain experts. In this case, scientists in terms of which entities, classes, and features should be selected. And working closely with domain experts, as you can see here, helps us solve some cool problems with very meaningful impacts. Thank you. You guys have questions?
Great. So we've got a question here from Kenneth. He said, great presentation and really interesting work. Just wondering how you deal with differences in experimental designs from various labs when using their data to build or validate machine learning models in Western blot digitization projects.
Yes. So like I mentioned, there is no standard way currently to do the quantification. So the most common way that after doing research we found was through tools like Photoshop kind of tools called ImageJ, where they draw a bounding box around the blot and then try to quantify that signal with an underlying background subtraction algorithm. And we tried to kind of mimic that and test that the values that they see is kind of we showed our outputs to scientists, and they kind of validated that these to a lot of scientists, and they validated that these values make sense looking at that image.